Ladies and gentlemen, please take your seats. Our program is about to begin.
Ladies and gentlemen, our program is about to begin. Please silence your cell phones or place them on vibrate.
أيها السيدات والسادة، سيبدأ البرنامج بعد قليل، لذا نرجو منكم التكرم بالجلوس في مقاعدكم.
Absolutely on the point.
I really enjoyed the environment, enjoyed the topics that they gave us.
Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. This is Farhan al-Shahrani. I'm moderating the session today. And I am very glad to be with you guys. And I'm so sorry that I'm not on the stage with you. I'm so sorry for that. But I was asked last night to stay home. So anyway, we will be using the technology. Hopefully, it will not fail us. Today is number 4 of the Winter Enrichment Program of KAUST, which is the theme is resilience, covering all aspects of research themes that is the focus of KAUST in water, food, energy, environment, health, and digital. Today, our speaker is Dr. Junaid Kashir from Alfaisal University. A little bit about the university. It's a private university in Riyadh, founded by King Faisal Foundation back in 2002, and started admitting students almost 12 years ago. So more or less is the same age of KAUST. And they are focusing on business, engineering, medicine, pharmacy, science, and general studies. Alfaisal University, as you know, it's a strategic partner of KAUST. A couple of months ago, we signed an MOU with Alfaisal University to kick off or launching the MD PhD program, which is one of its kind in Saudi Arabia. And I'm very glad about the explanation that Dr. Junaid mentioned this morning in his interview by KAUST people. He explained this program as an adapter to connect fundamental research with real medicine practice. So, and this is why we are delighted to invite Alfaisal University, and Dr. Junaid came forward to be here on the stage today. So we must thank him and Alfaisal University, and everybody is attending, whether in here or in the audience like myself. So Dr. Junaid, academically, he has a bachelor's degree in molecular genetics from the University of Sussex, followed by a PhD from the University of Oxford. And while there, he was a staff member also in the master degree program, clinical embryology program. After that, he got his PhD in 2012, followed by 2 years of postdoctoral research at the University of Oxford, as well as Cardiff University. Following this, Dr. Kashir had 5 years of health research fellowship with NHS in England, while being associated also with the Alfaisal University in Riyadh. And in Alfaisal University, he successfully established the master's program in clinical embryology and the reproductive biology. And this is a hint to his speech today. Dr. Junaid, and this is good news for all of us, was promoted just last week to be associate professor. And probably Web and Uncaused is the first platform to announce this. So congratulations, Dr. Junaid. So in addition to his academic research and what have you, he also embarked on several managerial positions: Director of Research in the College of Medicine of Alfaisal University, Director of Biomedical Laboratory, Chairman of Safety Committee, Chairman of Medicine Research, and more positions. So during his career, he published more than 50 internationally peer-reviewed, chapters of books. He's fellow of the Royal Society of Biology, as well as fellow of the Institute of Biological Science. Today's keynote speech by Dr. Junaid is very much aligned with the theme of WEB, which is resilience. Resilience in the spark of life, fertilization, calcium oscillations, and clinical perspectives. So he already sent me a brief of his presentation, but I don't want to steal the thunder. I will leave the floor to him, and the floor is yours, Dr. Junaid.
Wonderful. Thank you so much, Dr. Farhan, and thank you so much, everyone over here, for having me. It's something that I always say, that the beauty of this campus is only superseded by the generosity and the support of the people here. So thank you so much for every single visit. Thank you so much for all your support. So today will be a little bit of a different talk, I hope, a bit of a humorous take, I would like to imagine as well. And it mostly concerns reproduction. And the clinical aspects surrounding reproduction. And the reason why we actually chose this topic is you can't really get any more resilient than life. And within life, you can't actually get any more resilient than the way in which it reproduces. But before I go into that, it's, in terms of public speaking, it's always advisable to start off with some sort of joke to try and get the audience engaged. But considering that this is being live streamed on YouTube and the subject matter, which is reproduction, you're just trying to play it safe, don't want to kind of make any unnecessary jokes. So the next best option, if you don't want to make a joke, is to actually give a little bit of a fact or a little bit of a story about your background of yourself. And before I actually talk about the science bit, I actually wanted to talk a little bit about my own motivating factors as to why I came into this. And what a lot of people actually may not know, and probably they will now, is I actually spent a large part of my childhood here in Saudi Arabia. I grew up in Jeddah. And of course, like any adolescent growing up, you're consumed by pop culture. And one of the things that I was consumed by personally was both the movie and the novels, Jurassic Park. Of course, I'm not going to talk about Jurassic Park in itself, but essentially the resurrection of dinosaurs and creating a park, and everything goes wrong. But the key thing about this movie, apart from it being absolutely amazing, was the key characters in it. And I was actually really, really, really touched by the portrayal of this genetic engineer, this scientist, portrayed by the late Lord Richard Attenborough. I wanted to be someone like him. I wanted to be this suave yet eccentric scientist who, utilizing mosquitoes buried in amber, extracting blood from that they had taken from dinosaurs, what this person seems to be doing on the right. It just really motivated me to go into science. It's what motivated me to actually start to try and think about resurrecting dinosaurs, to try and come into this sort of field. And for me, it was a really proud moment, actually, when I graduated with my bachelor's at the University of Sussex. The chap who gave me my degree, because he was the chancellor of the University of Sussex, was this very same man. So it was a very rewarding experience for me, and it motivated me to go even further. Once you grow up as well, you find out a lot more facts, and you find out that actually the mosquito species that they used in the movie was actually the one type of mosquito that didn't actually consume blood. So, Hollywood not being exactly accurate living up to its moniker. So that really motivated me to think about molecular science, to think about how to utilize science for more practical aspects. And the second character from the same movie, who also influenced me to a great degree, was, of course, the great Ian Malcolm. And, this infamous picture here where he's kind of shirtless and musing on the kind of meaning of life. He kind of says something which really resonates, at least for me, with the theme of this particular web. Just life finds a way. You can erect barriers, You can create difficulties, you can try and stop, you can think that you stopped life in its tracks. But life is resilient. It finds a way. It manages to thrive. And what better example to demonstrate this than the way in which life reproduces? It's absolutely astounding the type of strategies that the various animal species, and non-animal species as well, but we'll focus a little bit for this talk, that the type of strategies that they come up with. Starting off with the humble Drosophila, one of the major developmental biology systems, the fruit fly. The way in which they reproduce, it's absolutely amazing. These are a polyamorous species. The female mates with multiple males. But what's absolutely astounding is that the female can actually store the sperm of the multiple males for varying amounts of time. And what's even more astounding is that the female knows exactly where, more or less actually, where each of the male sperm is stored. So when ovulation occurs, when it's time for reproduction, she can, on demand, release a particular male sperm. Absolutely amazing. And to counter that, to actually illustrate the battle of the genders, what the strategy males have come up with is that usually you heard about the race for life, sperm competing with each other. Well, in the Drosophila, sperm from the same male can actually cooperate and form this kind of super propeller sperm to try and outpace all the other male sperm as well. So these absolutely amazing strategies are also then demonstrated by another model. Over here, these are eggs of the zebrafish, Dania rerio. And they've actually evolved that instead of lots and lots of sperm trying to compete to enter the oocyte, what happens here is that a single sperm needs to enter this tiny hole called the micropyle. So only this particular strategy, which seems to be unique to this particular type of species. Again, another very different strategy that evolution has thrown up. And then just to illustrate further the differences, now let's go to mammalian species. So over here, you see on the left, this is human sperm. Compare that to this Captain Hook-looking fella, mouse sperm. Morphologically, very, very different types of cells, yet performing more or less the same functions, going through more or less the same processes, but looking very, very different. And all really geared to try and do this, the union of the two gametes, fertilization followed by embryogenesis. Now, just this process to get to this stage is a monumental task, and yet it's still not the end, it's just the beginning. There's so many things, so many barriers that life must overcome, and yet it does. In fact, the more you read about it, you really do realize just how much of a miracle all of us are. Any single thing could go catastrophically wrong, even the smallest mistake, even the smallest of mistiming. And given just how evocative this subject is, as a species, you can understand that we've actually been trying to understand this for a very long time. But it's quite surprising to know, actually, that in terms of modern science, in terms of trying to explain this, we've only relatively been trying to do this for a very short amount of time, in terms of experimentation, followed by outcome, followed by observation. So what I'm just going to do now, just to demonstrate this, because to know where we're going, you need to know where we're coming from, is to go through a little bit of kind of historical experiments that we've done. And these are key bits in the long scheme of things. So I'll start off first by this guy, Lazzaro Spallanzani, who was a major scientist within the field. And in fact, he was one of the few people who disproved a theory that was prevalent at the time, which was just spontaneous creation. So before him, as a species, we actually believed that life just appeared like this, that there wasn't anything that went on. And he performed a series of key experiments which actually determined that you need both a male and a female component. One of the experiments that I like to talk about is something we call frogs in pants. And it just demonstrates, the resilience that scientists also have to demonstrate to be able to demonstrate particular outcomes. So what he did was he took two groups of frogs, and he's mostly associated with frogs. There's a statue of him holding a frog as well. And one group, he didn't do anything. He just unleashed onto females. And of course, what happened, they mated, and they were able to result in tadpoles. The other group, though, what he did, demonstrating just how much time they used to have back in the day, he made them wear these tiny rubber pants. And then he unleashed them onto the females, and of course, there weren't any tadpoles that were produced. So really, experimentally, is the first time he established that sperm was a requirement. And he also established in many other ways that you also needed a female component as well. But really, at the time, you also have to remember, culturally, there are certain things which were acceptable and non-acceptable. And what people seemed to latch on at the time was, well, no, it's the male component which is the most important. So yeah, well, let's discard the spontaneous creation theory and say, well, okay, no, the male has a divine right to create life. Moving a little bit forward now, what a lot of people actually also don't realize is reproduction as a field is quite heavily dependent upon microscopes. And it's very fitting that when the first microscopes were being invented and being refined, the very first thing that was observed on the microscope was pond scum, so pond water, bacteria and the like. The second thing was human sperm, as you would. And the observations that were made were quite striking. They allowed scientists to come up with some weird and wonderful theories, but if you look at the accuracy of the drawings, they're not far off compared if you think about the primitiveness of the actual equipment. The very first theory that was proposed was the Homunculus Theory, which stated that the sperm was almost like a vesicle, and it carried a miniature version of the organism which was delivered to the female. This was then further refined to say that the sperm itself was a miniature version of the actual organism. So, it's really astounding the kind of directions that we're coming from. And yes, with every single advance, we make more and more gains in terms of knowledge. Well, let's fast forward a little bit more to basically get this entire sperm-dominated picture completely turned on its head. Let's go to the start of the 19th century, and along comes this chap here, called Jacques Loeb. And he did a series of very key and very elegant experiments. He took sea urchin eggs, which are transparent, which also allows them to be utilized as a developmental biology model. And just by changing the concentration of the salt in the water, he was able to induce embryogenesis. No sperm required. So just by changing the concentration of the salt, the constitution of the actual seawater, he was able to induce embryogenesis. And of course, this, we don't, as a species, we seem to only really work in extremes. We go from one extreme to a complete other, that female involvement is not allowed or not actually important, to going to a scenario where we think that actually the sperm isn't important. And this led to a lot of really funny stories that, we're going to a world without men, and also many stories of a seaside holiday resulting in magical pregnancies by bathing in the water, which is very relevant since we're close to the sea. So it's just absolutely amazing where we've actually come from. And of course, now we know, or at least we think we know, what the basic requirements are for reproduction. And a lot of the work that has been done before has led to the modern scenario where we're actually able to treat cases where this process goes wrong, particularly within humans. And that's where my talk actually comes in. What I'll be trying to do is to relate to you the growing problem of infertility worldwide, considering that we're still going through what's been a devastating pandemic. One should like to think that you want to be prepared for the next sort of medical emergency. And believe you me, this is one of those ticking time bombs, just given the numbers that I'll be talking about. And then moving on from that, I'll be also utilizing some of our own research as well as purely as an example to illustrate to you the type of approach that we're taking to try and improve cases of success in terms of fertility treatment. So just to kind of start off with, definitions are a big thing, but it's important to actually mention that infertility itself is classified as a disease by the WHO. The actual details of this can differ in terms of the amount of time that's required for you to be classified as infertility, but classified as a disease of the reproductive system nonetheless. And there's a reason behind this. Like I mentioned before, the numbers are absolutely astounding. Globally, and these are relatively older numbers now, actually, the real figure is probably a lot higher than this. 1 in 7 couples worldwide will be outright infertile. On top of that, on top of the 1 in 7 figures, men, unfortunately, guys, we have it a little bit more rough. because 1 in 5 men will be subfertile, so not infertile outright, but will have difficulty in conceiving. So these are just absolutely astounding numbers. If you think about the sheer scale, the sheer population, some of you might be thinking, well, there's so many people, there's never been any more people alive at the moment on the planet than ever before, but this is a compounding problem, especially considering that many more people are now seeking treatment for such cases. Now, it then becomes necessary to try and dissect what's causing infertility. Only if you can appreciate what the cause of infertility is can you then try and treat it. And really, when you try to break it down, you realize that infertility, like many other disease conditions, is very multifactorial. It's not just one particular reason, there can be multiple causative factors. But generally, you can actually split it into male versus female problems. Now, in female issues, the issue is much more diverse. You can get many more types of different cases. Of course, if you exclude lifestyle choices, environmental factors, as well as societal issues. Predominantly, though, in terms of male infertility, it seems to be mostly sperm issues, or either an insufficiency of the sperm or a failure to produce sperm. Now, we were just earlier on, we were having a similar debate as to what is causing, what the proportion is in terms of male versus female. And really, if you ask, it depends on who you ask. If you ask a medic, they will automatically say it's about 50-50, 50% female, 50% male. But those of us who are on the front line of research can actually quite safely say, well, quite by a large margin, it seems to be male issues. Because the reason for that is a large chunk of the pie, as you can see over here, is unexplained infertility, where we know that there's an issue, obviously nothing's happening, but we can't really explain what's going on. And the reason why male infertility kind of ekes out as a leading causative factor is because the majority of these cases seem to have something wrong with the male side of things. But you see, this entire slide that I've just talked about is somewhat irrelevant as well. Because the first mistake that we do when approaching this is we try to find causative factors. We stick to the black and the white. What we actually should be trying to do is viewing each of these cases as contributory factors. Just as there's a saying, you can't clap your hands with just one hand, although I'm sure some people could, but just generally, if you bear with me, it's not that there's a black and white. There will be cases where there's an outright individual problem, but generally speaking, most cases of infertility will have contributory causes from both genders. And then add on top of that environmental factors, diet, and lifestyle. It really is a very devastating picture indeed. And because of this growing issue, or at least in response to this growing issue, a series of laboratory techniques, a series of clinical techniques have been devised to treat the various forms of infertility. And these are actually based in the laboratory but utilized in very clinical ways, so very basic science being applied in a clinical scenario. And generally speaking, assisted reproductive technologies, or ART, are relatively quite good at treating infertility. Around about 70%, 65 to 70% of cases can be treated by ART. And just to give you an idea of how much this area is booming, as of 2012 at least, 4 million babies in the world were born as a direct result of ART. In fact, in some developed countries, 7% of the entire population is as a result of ART. Now think about that for a minute. 7% of populations of entire countries began life in a dish. And that number is only growing. When we talk about ART, or assisted reproductive technologies, you need to understand what it's meant by a particular round. Because it's not just one particular process, it's a series of collective processes. The first part, which you could divide it up into, is obtaining both gametes, both the egg as well as the sperm. And of course, there are various ways, various conditions, various considerations that need to be taken for this, but generally, you need both gametes to start off with. The second part is you need to combine both of them. Of course, easier said than done. And it very much depends on the type of infertility, which will determine what type of method you use to combine them to create your embryos. The final step, then you need to decide which embryo is good enough to then transfer back to the uterus. And then after that, beyond our hands. Still 50-50, whether or not it implants or whether or not it's abortive. And we'll talk more about that later on. But really, ART, as I keep mentioning, is more of an umbrella term. It's not just one particular. Commonly, in terms of popular science or popular opinion, one would recognize ART as IVF. And technically, yes, ART is IVF because it's fertilization happening in vitro inside a dish. But there are multiple technologies that constitute ART. One of these, of course, cryopreservation vitrification, mostly used in fertility preservation. It's actually being utilized for much, much more than just fertility aspects as well, but it's a very important technique nonetheless. You have other more advanced methodologies, such as embryo biopsy, where essentially one is able to take an embryo and either slice out a bit of a tissue from that, or even an individual cell. And these are then utilized for a very advanced series of genetic testing, prenatal genetic screening or diagnostics, to try and isolate or diagnose genetic conditions in embryos to perhaps prevent transmission of genetic disease. You also have many more advanced methodologies which we're improving as our understanding of reproductive biology increases, such as in vitro maturation, whereas you take immature eggs from within the ovaries and you mature them within a dish. So there are many, many more of these as well, but one other technique which I would like to focus more on, especially since by far this is the predominant method that's used within fertility clinics worldwide, increasingly more so, something called intracytoplasmic sperm injection, or ICSI. And essentially, it involves quite a lot of things, actually. And I'd like to give a shout out to Muhammad Rajab, who is our beautiful model here. Very, very photogenic. Thank you, Muhammad. And essentially, this is basically a microscope with a few additions on it. And these additions are essentially injecting capillaries. On the one hand, you will have a holding capillary. This will carry a hold of your oocyte, your egg. And the other capillary is just big enough to be able to take a single sperm. And utilizing this setup, once you take up the single sperm, you then inject it directly into the oocyte. And this is just a video showing you this happen. This is courtesy of the Oxford Fertility Unit, so thank you for providing this. And this is a very powerful technique, as you can see. You take a single sperm and inject it directly into the oocyte. This surpasses a loss of the barriers that are experienced during infertility, so you pass a lot of the oocyte barriers. You can get past many more male factors, so for example, immotility, abnormal morphology within the sperm. So the issue, though, is what happens when this fails? What happens when you bring the camel to the water and the camel doesn't want to drink? And it does happen. And it's not just in this particular case. There are many other types of scenarios where infertility fails, or infertility treatment fails, rather. Now, I've spent a large part of the talk actually, preaching about how good ART is. I'm just going to now turn this on its head. It's a powerful set of techniques, yes, but success rates are, quite frankly, pathetic. The success rate in terms of take-home pregnancies rarely exceeds 30%. In fact, even in terms of fertilization, even these powerful techniques, such as ICSI, they rarely exceed even 24%. Now, this is per cycle. Now, when I say per cycle, it's this entire, the steps 1, 2, and 3. That constitutes at least more or less one cycle. In fact, predominantly, for a successful round of fertility treatment, one needs, on average, 2 to 3 rounds of fertility treatment before a successful pregnancy can be established. And that's a problem, especially since the success rates are so low. It's a problem because not only is this financially demanding, even in states such as the UK, for example, where the state covers a large part or shoulders a large financial burden, it can still cost quite a lot of money, around about $6,000 to $15,000 per cycle, depending on what type of treatment is needed. But then there's also the huge emotional cost. Now you have to remember, considering which definition of infertility you use, to get to the stage where you're actually being allowed to use fertility treatments, you're emotionally exhausted already. And this is your last option. And then when you fail this again and again and again, it can be absolutely devastating. So what we need to really try and do is to improve this scenario. And the only way in which we can do that is improve the methods, improve the users, and improve the science. Because really, despite all of these, and you'll see these figures again and again throughout these talks, ART is here to stay. Successful. It's being used a lot, increasingly ever more so. So one of the things that needs to be done, of course, is education, intensive training. And this ties in with the collaboration between Alfaisal and Kaus in terms of the MD PhD. Because to be able to do ICSI, for example, everything you see in this picture, Muhammad included, Muhammad is optional, of course, we keep him for ourselves. Everything you see in this picture is a requirement for a successful ICSI. The operator, yes, and not just any person. This person needs to be trained in reproductive biology. This person needs to have clinical knowledge. This person needs to know the science behind the technique that he's doing. He needs to understand he or she needs to understand gamete biology, embryology. But this person also needs to be competent in handling this equipment. What if something goes wrong? What if there's a leak? What if there's some sort of error? Are you going to wait for an engineer to come and repair it for you? You have someone's gametes on your microscope. You don't have time. Everything is being handled by this one individual. The fire extinguisher as well, because you're responsible for the safety of the lab. So this really just states and demonstrates my point that not just anyone, you can't take any random person on the street, put them inside a lab, and then expect them to be good at it. And a large part of this is something that we've been involved in. I've been involved personally, both at Oxford, as mentioned by Dr. Farhan, as well as here at Alfaisal, where we're actually training not just the embryologists of the future, but also in the mind of training the next generation of reproductive biologists and scientists. Because it's a holistic understanding that's needed, not just a clinical scenario. And these efforts are in part driving a lot of research, a lot of advancement in terms of embryology. One of the things, of course, here is that's being utilized more and more so is something called sperm DNA fragmentation, where we've essentially found that the DNA inside the sperm needs to be in a certain state. If it's damaged in some way, i.e. fragmented, then it won't be able to result in successful embryogenesis. And that seems to be a leading cause behind at least male infertility. And we're coming up with novel techniques to be able to detect these sperm and treat accordingly. We're also utilizing time-lapse microscopy, where essentially we make movies of developing embryos and look at the cell dynamics. If the cells are dividing, if embryogenesis is proceeding too fast or too slow, it's not going to become a good embryo. So these technologies are increasingly being utilized. And of course, we're adapting novel technologies as well. Next generation sequencing for PGS and PGD. So in a very large part, ART is serving to really help the development of a lot of novel technologies. And what I'm going to do now is to talk a little bit more about my own research to demonstrate to you the approach that a lot of people are taking. Before I do that, actually, I wanted to also demonstrate the extent of the applications for ART as well. Something called three-parent babies. You might be familiar with it. It was all over the news a few years ago. And essentially, this has to do with something called mitochondrial disease. So mitochondria being, of course, the battery, commonly known as the battery of the cell. The vast majority of mitochondria in our bodies are inherited from our mother. And this is demonstrated actually from this picture over here. In red, you can see all the mitochondria within the oocytes. The sperm also delivers a little bit of mitochondria, but it's negligible, and we don't actually really know what effect it might be having, if any. But the issue then arises because mitochondria has its own set of DNA. In fact, that's how lineage tracing is done. You're using mitochondrial DNA markers. And this unique set of DNA is also responsible for a number of biological processes. So if you have mutations in these, you will get, of course, disease. So one of the things that people have tried to use to solve the issue of mitochondrial disease is, of course, surrogacy, treatments, of course. And, you know, it's even with treatments prior to this, it's only really prolonging the issue. You're not really solving it. So utilizing ART methods, of course, pioneered in the UK, we're looking at transfer of genetic material from one oocyte to another. So the way in which this is envisaged is basically you have an affected oocyte from the mother, you use a sperm from the father, fertilize, and you create this zygote. But of course, the mitochondria in this will be affected by the disease. You take out, after this, once the fertilization has happened, you take out the DNA and transplant that to a donor oocyte. So this is an oocyte from a third individual whose DNA has been removed, but the mitochondria is still there, which is also genetically normal. So that's where the moniker of three-parent babies come in. And it's just demonstrating how ART is expanding in its scope as well, being utilized for other types of medical conditions. And of course, you have many other applications which we'll talk about very, very briefly towards the end. But the issue has always been that advances in reproductive biology, or indeed ART, have been very much individually focused. Either you're focusing on the sperm or the oocyte, or you're focusing on the embryo. And what we're realizing now is that, well, actually, you need more of a holistic approach. You need to be examining all aspects of this particular problem. And this is where I'm going to now talk a little bit about my own research to demonstrate how we're trying to do this. I'm not claiming that we're doing this properly, but we're trying to at least. And this has to be able to explain to you my research, I want to give you a little bit of background about it, and it leads on from the experiments that Jacques Loeb did in terms of changing the salt concentration, which caused the embryos to start developing without any sperm. In fact, this is not really a miracle. It's a common phenomenon that we know. It's something called parthenogenesis. And it's induced by excess amounts of a particular ion. And to cut a long story short, at the moment of fertilization, the oocyte is basically a dormant cell. And it needs fertilization by the sperm to be able to start developing into this particular embryo. So the sperm, if you want to use analogies, the sperm being Prince Charming, needs to awaken the Sleeping Beauty, which is the oocyte. And it needs to do this in some sort of signal. It needs to tell the oocyte, hey, it's time, I'm here, we need to start combining now. And the way in which the sperm does this is that it causes a large increase in the amount of calcium. Now, of course, calcium, very, very busy ion. This is just a part of all the different signaling pathways biochemically that calcium is involved in. Ultimately, it's involved in basic processes such as muscle contractions. It can control which genes are being expressed or which are being shut down, and ultimately even controls the fate of a cell, controlling cell death. And its importance isn't lost during fertilization either, because another series of really key experiments utilizing something called calcium-specific dyes, so these are dyes that give off light in the presence of calcium. These really show to us what calcium is doing. So this is a fertilizing Medaka fish egg, which has been injected with this calcium-specific dye. So the more calcium there is, the more light will be given off. And as you can see, from the pole of fertilization on the left-hand side, you see this huge wave of calcium just traversing from one pole to the other. So in some species, this is the case that you get this single transient increase in levels of calcium within the oocyte. Mammals, though, are a little bit more complex. Instead of a single transient, you get multiple transients. So this is a video of fertilizing mouse oocytes, which have been injected with a calcium-specific dye. This is courtesy of Professor Keith Jones. Thank you very much for this video. And as you can see, blue levels indicate resting levels of calcium, and the redder it gets, the more calcium is present there. So instead of a single increase, you can see these are turning on and off like Christmas lights. So instead of a single transient, mammalian oocytes undergo calcium oscillations. The levels go up, they come down, they go up, and then come down. And this repeats for a certain amount of time. Now, it's not that they repeat for a certain amount of time. It seems to be that each single species has its own unique pattern. Even beyond species, but within the same type of oscillatory species as well. So in some species where you have a single transient, such as, say, in this example, sea urchin or xenopus, they both have a single transient, but the way in which this calcium is released is very species-specific. Even in species where you have multiple oscillations, say, for example, ascidians as well as mice, the profile, the pattern, the amount of calcium, the timing of these oscillations, they're very, very unique to each species. It's almost as if it's some sort of species-specific signal. In fact, this could be one of the reasons why chimeras are so notoriously difficult to make, because each of them have their own unique biological signatures. Of course, there are many other issues involved, but this could be a leading issue. In fact, so important is this profile of calcium release that they actually affect the profile of gene expression. If you were to take the same embryo with the same sperm, but elicit different profiles of calcium release, you would get completely different gene expression profiles. So these are very, very powerful signals right at the start of life. It's almost like the domino effect. So you line up these dominoes, and how these dominoes fall very much depends on how it all starts. If you remove a key piece of the puzzle, everything just starts falling down. Now, the key thing that we've been working on for the past decade or so is a sperm-specific protein. So it's only found within the sperm, which causes these calcium oscillations. It's an isoform of PLC, phospholipase C zeta. And it's a very, very unique isoform of the 13 known PLC isoforms, at least within mammals. It's uniquely located within the sperm, at least as far as the literature is concerned. And it's found to be testis-specific. It's found to be in a number of different species, mammalian and non-mammalian, indicating perhaps a universal vertebrate mechanism. Importantly, if you deplete sperm of PLC zeta, you result in infertility, particularly within humans. In fact, you could also utilize PLC zeta as perhaps a potential diagnostic and therapeutic, importantly, for cases where this protein is insufficient. So this really kind of demonstrates a very elegant mechanism, a very elegant model to explain fertilization. In fact, it still is a little bit controversial as to what the identity of the factor that causes the calcium oscillation, but the vast majority of the field agrees that at least the major signals for the calcium release are because of PLC zeta, where essentially you have the sperm fusing with the oocyte. This releases PLC zeta into the oocyte, causing a signaling cascade and resulting in calcium release. And it's basically utilizing these same principles that we've actually envisaged in the field that we could try to use PLC zeta as a therapeutic, where in cases where sperm is defective in this particular protein, we could inject it with the sperm. And this is a series of key experiments that we did way back in Oxford, where essentially we took RNA encoding for fluorescently tagged PLC zeta. Yeah, that's right. We've been shooting stuff up with RNA before it was cool, as it is now. And it works in principle. Essentially, you add the RNA, you inject it into the oocyte, and the transcriptional machinery within the oocyte will make the protein for you. And of course, RNA is perhaps not the most efficient way, which is why we've been trying to make recombinant protein versions of this. So what we've essentially been doing is to try and understand these calcium oscillations and how they're impacting embryogenesis. And of course, in a very clinical context, how they're affecting fertility success rates. So to try to understand these calcium oscillations, we need to try and understand PLC zeta within the sperm. So we've been trying to characterize PLC zeta within the sperm, performing a lot of clinically relevant studies and trying to utilize this both as a diagnostic as well as a therapeutic, and perhaps to try and explain other cases of infertility as well. Some of the most recent findings that we have are really actually suggesting that the amount of this protein that's delivered has a direct impact on the quality of embryogenesis and success of pregnancy as well. And actually, studying this protein and studying the mechanisms behind this have also revealed quite a lot of basic mechanistic biology that we actually didn't even consider, such as the involvement of nanovesicles. Now, in terms of nanovesicles, this is something that has been completely unexpected for us, at least. And just to kind of explain these, any given cell is just chucking out nanovesicles. You might know them as exosomes. You might know them as vesicular components, liposomes, for example. But the idea is that these are vesicles on the nanoscale. And any given somatic cell is chucking these out in the order of millions per hour. And the reproductive system in mammals is no different. A really important tract within the male reproductive system is, of course, the epididymis. This is secreting nanovesicles. The prostate, that's also secreting nanovesicles. And within the female reproductive tract, the uterus, as well as the fallopian tubes, they're also secreting nanovesicles. Now, what's important about these vesicles, at least as far as the sperm scenario is concerned, these not only interact with the sperm, but they also deliver RNA, they also deliver protein, they also deliver DNA. In fact, such is the impact of these that a few studies back in 2016 published in Science demonstrated a solution to a very unique phenomenon that we used to observe within reproductive biology. And this phenomenon was that if you took mice, specifically male mice, and you fed them a high-fat or a low-protein diet, the offspring of specifically the male mice were metabolically abnormal. Decreased glucose metabolism, autism-like defects, stuff like that. And again, stressing that this seemed to be a male-specific thing. These mice were genetically completely normal. They were outbred, no inbreeding going on. And we just couldn't explain it as a field. We looked at all sorts of different epigenetic mechanisms, long non-coding RNA, different types of proteins, everything you could name. And it wasn't until 2016 that we realized what was going on. It was discovered that the sperm of these mice were enriched in a type of RNA called tRNA fragments, quite literally fragments of tRNA. And these tRNA fragments were essentially what were causing these metabolic abnormalities. What was unique, though, is that these tRNA fragments were being delivered to the sperm via nanovesicles. So one just thinks, or perhaps imagines, that there is a scenario where you could utilize these nanovesicles not just as a therapeutic, but also as a diagnostic for many other purposes. So this particular process that I talked about called oocyte activation, a holistic overview of this process is not just indicative of fertility, but there are many other implications for it as well. To understand embryology is to try and understand stem cell biology. To be able to understand that, you also understand cancer biology. Indeed, genetics and epigenetic remodeling, that's something that is pretty major at fertilization. That's something that our research is also touching upon. Biomechanics, the sperm motility, the corkscrew motion of the flagellum, for example, that's being utilized to create micro-robots, for example, propellers. Neuroscience, now that's something that's a topic for a separate discussion, but essentially it's very astounding that gene expression of testicular proteins seem to be matched with neural expression as well. And of course, you have technology development. ART is one of those fields which is really driving novel technology applications. One of the most recent advances is microfluidics, to try and select the best quality sperm in the most natural way possible, as well as oocytes as well. Now, the reason why this is important is because ART, like I keep on saying, isn't necessarily a silver bullet. In fact, it might even, because we don't really know what we're working with, it might even be causing more problems than solutions. So a lot of studies, again, this is very, very controversial. Some studies say that there aren't any effects, but some studies are saying that children born via ART have more issues with their fertility compared to their normal counterparts. Now, one would expect, well, of course, because they were born from an infertile genetic landscape, it would make sense that, of course, they would suffer from similar conditions. But if you have a treatment in the form of ART, does it really matter? The real concern seems to be the potential link between ART and epigenetic disease. Multiple conditions are now being increasingly associated with ART in terms of epigenetic factors, but we're still not certain whether ART is causing these or whether it's the other way around, or whether it's just by chance that we're seeing this in terms of a population scale. One thing is certain, though. ART is becoming an increasing requirement for our society. In fact, a very large study looking at most Western clinics has found that over the past 50 to 60 years, at least in the male side of things, sperm parameters have fallen, i.e. sperm has become less fertile by around about 50 to 60%. Now, could this be because more males are attending clinics? It's becoming less of a taboo. Could it be that we're trying to characterize better now in terms of what these conditions are? The fact of the matter is, ART is here to stay. It's successful. It's utilized in a very widespread manner, but because of this, it's also big business. Now, you don't have to look far. Let's look at the two most populous nations on the planet. If you look at China, this is the biggest market for ART right now. Close to around about 1.5 million cycles a year, and these are only a very small subset of clinics reporting their figures. In India, reproductive tourism, that's a big thing. Even there, around about 250, 300,000 cycles a year. If you consider that each cycle is about $6,000 to $15,000, it's a big business, really big business. Now, we can do one of two things. Either be very much opposed to this technology and just basically advocate, well, only use it where it's needed and shun it where it's not. Or the approach that I personally like to take. Admit that, yes, improvements need to be made and make them. But the only way you'll be able to do that, understand that you have a problem, try to solve the problem by trying to understand the science. And that's going to be the end. Just before I end off, just coming back full circle, you might have all seen on the news that this perfectly preserved dinosaur egg was discovered, as published by University of Birmingham. And really, this demonstrated the current theory here that the dinosaurs didn't actually just die out. They basically, because of certain environmental factors, whatever they may have been, they actually evolved into some sorts of birds. Now, because I started off by talking about what drove me into this field, I couldn't actually, I've not been able to make dinosaurs. I ended up making human babies instead. But with some of the studies that we did, considering a link between birds and dinosaurs, and considering that at least my PhD supervisor, one of the key things that they did was they discovered that even chicken had PL-Caesisa. The dream lives on. Dino PL-Caesisa? Question mark. Thank you so much, everybody, for your time. I just want to thank all of these people. I first started to type out all these different names, but it was way too many people to fit on the slide, so I just put on all of these affiliations. Predominantly, I want to thank Alfaisal University, of course, for giving me the platform that they have, the funding bodies, CACs, and of course, predominantly you. Thank you so much for your time. Thank you so much for being the audience today. I'll take any questions. Thank you so much.
Thank you so much, Dr. Junaid, and again, thanks to you, to Alfaisal University, our partner, and having the trouble last night to come to the stage.
My pleasure, absolutely.
I received already a few questions from the audience and in front of you, as well as some through Zoom chat. And I'm very happy to hear about the history of fertilization, the cell dynamics. The theories, some of them artificial, controversial for sure. And when you mentioned the technology of ART and for million babies, started their life on dish, it's an amazing. Probably as your approach is moving forward and probably making sure this works perfect for everybody, as it's the case now with China and others. Dr. Junaid, I summarized the few questions, some of them very much related to the talk itself, some about Alfaisal University, its future outlook, and the web itself. So if you allow me, I'll start with the talk itself. Fertilization is a big subject, and it's extremely hot topic in some countries in Europe and elsewhere, and probably so in Saudi Arabia and the region. What is happening? What is the situation in Saudi Arabia about research centers related to fertilization? Is there any statistics? Where are we heading? Can you tell us, please, more?
Well, certainly. I think actually, one of the key areas that I'm interested in actually is to try and develop the landscape in terms of fertility treatments within this region, because there's actually a whole series of issues that are unique to not just Saudi Arabia, but the GCC region in general and the developing world, particularly North Africa, the Middle East, as well as the Asian subcontinent. Now, in terms of the direction of travel for fertility treatments globally, it's always been kind of something called elective single embryo transfer. Now, the name's always changed throughout the years, but what the field is aiming towards is essentially a scenario where you can select a single embryo and be sure that that's absolutely perfectly developmentally competent and transfer that, and be sure that it's going to result in a pregnancy. Now, people have tried various aspects, technological advancements, but really what's really held us back, more than the technological capabilities, is our lack of understanding of reproductive biology and reproductive sciences. Of course, this situation is changing. As more and more research becomes more holistic, as I told you about, we start to move away from one particular model. But there is still a long, long way to go. And as I mentioned before, a large part of the solution is adequate education. And that's one of the things that we're a key proponent of and something that we're advocating quite strongly. In terms of Saudi Arabia, in terms of statistics, I'm afraid there's not much by way in terms of official statistics that one could quote. There are relatively few studies within Saudi Arabia, and it's predominantly due to the developing infrastructure. And of course, the situation is indeed improving, but generally speaking, it's quite reflective of the global scenario. But in terms of what we can say for sure in terms of the popularity of ART, big business. Just in the United Arab Emirates, within the space of 3 to 4 years, you had something like a 350% increase in the number of clinics opening, as well as the number of cycles that were being performed. So definitely the direction of travel is to increasing volumes, but to what degree we're not really actually sure. So hopefully as we develop more in line with the various governmental aspects, that problem will be solved.
Excellent. So I just received another couple of questions, one related to Alfaisal University. Since we are talking about resilience, and which is the theme of the web, how did Alfaisal University cope with the pandemic itself? This is question one. And how is your relationship to King Faisal Hospital and Research Center?
Well, actually, I think, Alfaisal coped just around about in the same way as KAUST did. There were many different initiatives, both by the leadership of the university, but also predominantly with a lot of governmental and ministerial support. So more or less as soon as the pandemic started, there were a round of funding opportunities available to promote COVID research. Of course, you had lots of other issues that were in place. Lots of committees were formed, safety committees, safety of both students and faculty was, of course, paramount. But really, a lot of effort and painstakingly so was taken to ensure that while the safety and while the social distancing aspects were kept in place, solutions to the problems that we had were continuously tried to be sought out, both at the research level, and I think our faculty responded brilliantly. I was actually a key player in trying to allocate funding. We had a competition, and we ensured that a lot of projects were actually funded. A really key success story is Professor Ahmed Al-Jada, professor biochemistry. He came up with a rapid PCR test, low-cost rapid PCR test as well. So that's actually in collaboration with industrial partners as well. So it's just one example of many other things. So me personally, we published a lot of papers as well, very early on advocating multiple alternative technologies for rapid detection. So in terms of response, I think it was a very robust response. Things could have been different, yes, but it was the first time that everyone was experiencing this sort of stuff. Now, to answer the second question, I think this feeds in with the resilience of our response to the pandemic is our partnership with the KFSH. A lot of people don't actually realize this, but initially, the College of Medicine at Alfaisal was envisaged to be the academic arm of the KFSH. And it's evolved into perhaps a better relationship, a more deeper relationship at the moment. And quite frankly, at the College of Medicine, we view the KFSH as our teaching institute, teaching hospital. It is the clinical side of the College of Medicine. And, you know, the partnership between the KFSH and Research Center, as well as Alfaisal, especially College of Medicine, runs very, very deep. I am a very good example of that. I work with Dr. Abdullah Asiri, Department of Comparative Medicine, and also Director of the Animal Facility. If you're watching, hi guys. So, you know, together, both in terms of research, we have a lot of papers that we've published, a lot of research that we're working on together, but also academically speaking. So I am the program director for the Masters in Clinical Embryology and Reproductive Biology track, and Dr. Abdullah Asiri is the co-director. And this is a very unique partnership that we have, both clinically, academically, and in terms of research. So very, very in-depth relationship.
Excellent, excellent. And at the same here, you know, we'll be delighted to have Dr. Majid next week as a keynote speaker of the web. At the same time, I think we are coming to the end of this session. I must thank you, Dr. Junaid, and we will be handing over a token of appreciation. I will call my colleague, Dr. Huda, to hand it over to you. Please. And with this, thank you so much, Dr. Junaid. We thank also Alfaisal University and its leadership.
Thank you so much.
And all the best. Thank you so much.
Thank you so much. Absolute pleasure. Thank you, everyone. All the best, and wish you all the best for all your future and your careers. Stay safe and stay happy. Thank you.